Thank you, and thank you to Philippe for this very kind invitation. I think it's a great honor to talk at the Collège de France. And today I will talk about IL-2 production by dendritic cells. But first I would like to really talking about dendritic cells to acknowledge Ralph Steinman, his contribution to this field. He discovered dendritic cells in the 70s, and he has been for us a great mentor and a colleague. And I really learned from him many important things, but also if you wish to get at the top of the mountain, you really need to have persistence and determination. And this is a very important drive. He discovered in the 70s that dendritic cells were kind of cells that would be specialized and will be very potent in initiating immunity. But at this time, really, nothing was really known about the molecular mechanism. But he was able to predict that dendritic cells will prove to be a critical accessory cell required in the generation of many immune responses. And at that time, and for many years, people did not really believe that dendritic cells would be any different from a macrophage, but he was very persistent. And I should say that the Nobel Prize that was given last year was really a very important recognition to the fact that really dendritic cells play a very important role in activating the adaptive immunity. We know so much about this interaction between DCs and T cells. We know that there are a series of signals that are very well documented and described. Signal 1 through TCR and MHC, signal 2 through co-stimulatory molecules. But we don't know much about signal 3, which is the cytokine that dendritic cells will transcribe and produce once they are activated by microbes. And of course, microbes is an entire word, and this single cell is capable of discriminating what kind of signal they get from the environment, and it's capable of signaling and translating this into something that leads to the initiation of the adaptive immune response. So this cell is really a key cell in bridging together both innate and adaptive immunity. And in 2001, we published this observation that once triggered by microbial signal, dendritic cells will produce IL-2, and that this IL-2 was instrumental for the priming of T cells. We know that IL-2 is key for the activation and proliferation of T cells. We know that both IL-2 and CD25 expression are induced by T cell activation, but the present paradigm is that this IL-2 is made as an autocrine loop, and that in vivo this is self-sustained, and the role of dendritic cells was not really revealed for many, many years. We also know that IL-2 does not have only a role as an immunostimulatory molecule, and we know that it can induce, of course, all these functional activities, but we have also learned that IL-2 can be also an immunosuppressive molecule, and most important maybe that IL-2 is a key molecule in the activation of T regulatory cells, and therefore has a role as an immuno-regulatory molecule. So it's really quite something for a single molecule, but maybe what we do not appreciate is the kinetic of the production and also the amount of these IL-2 secreted by immune cells that maybe can explain in a better way all these different functions. So I would also like to say that Charlie Jane, we understood very early that immunity starts at the host environment interface, and that these pattern recognition receptor, PRR, are very important to really polarize and induce in dendritic cells the kind of reprogramming that is needed in order to bridge the innate response with the adaptive response. And in reality, for a long time, these receptors were not clearly identified, and it took 20 years after the discovery of dendritic cells to recognize the first innate receptor for microbial ligand that will determine this ability of innate cells to translate a signal to the adaptive immunity. And I would like to mention, because this is quite recent, that indeed Ralph got the Nobel Prize at the same time as Bruce Beutler and Jules Hoffman, and that Bruce Beutler was the one that really identified the TLR that play an essential role in host recognition and response to microbial product. And in this paper in Science, where he was looking at the mutation in the TLR4 gene, and we were very happy to collaborate with him at that time. He was able to formally prove that the mutation would block the signaling and would, and then no cytokine production would be induced. So now we know a lot about all the repertoire of receptors that are present on dendritic cells, and we know that the TLR4, of course, is the one that translates the signaling from LPS, but also we spent some time with Francesca Granucci, my former collaborator in Italy, to look into this molecule because it also binds LPS. And indeed, we have found that in dendritic cells, not only the NF-kappa B pathway and the transcription factor can be translocated to the nuclei upon ligation of TLR4, but also that there were other transcription factors that were very important in the activation of dendritic cells. IRF3, of course, leading to interferon production, but we have documented that also NFAT is another transcription factor that is translocated to the nucleus and induce the transcription of many genes, including IL-2. And more recently, with Francesca Granucci, we published this finding that CD14, by binding LPS, is capable once it is localized in lipid raft, because as you know, CD14 has no signaling tail, but when it is localized in lipid raft, there is a kinase that activates PLC gamma 2, and PLC gamma 2 is instrumental for the calcium influx into the dendritic cells, and this calcium is activating the calmodulin and calcineurin complex, leading to the translocation of NFET, the phosphorylation and translocation of NFET into the nucleus. This NFET activation is a new finding, and we have looked also more recently during hematopoiesis if this transcription factor is also used, and we have found that yes, indeed, the myeloid cells use it a lot. So the question that we were addressing is regarding the functional activation of immune cells via these dendritic cell-derived IL-2. And we have been able to demonstrate both in vitro and in vivo in the mouse that, yes, these dendritic cells, these IL-2 produced by dendritic cells is activating NK cells and also naive cells. But we know now that in addition to CD14 and TLR4, there are also other receptors that are quite important in transducing signals that leads to calcium flux, and these are the dectin-1. And dectin-1, as you know, is a very important receptor that can bind fungi and fungi product, and curdlan or yeast are also binding dectin-1. And through this dectin-1, SYK is activated, and through SYK we have also this calcium flux. So there are a number of pathways that are used, and we were asking the question, is this IL-2 production shaping T-cell polarization? And T-cell polarization is now really a very important question and issue, because most of the disease that we try to understand, like inflammation, allergy, or even immune suppression, are really the result of a certain T-cell polarization. And we know on the T-cell side that there are a number of transcription factors that are induced and are responsible of this polarization, but we don't know much about the signals that are provided that are occurring between naive T cells and dendritic cells. And of course, because there are many cytokines that are produced, it's not easy in vivo to deal with this question. But we made in these few years a number of observations, and I would like to show you some old data that may teach us something. For instance, that this transcription of IL-2 that was revealed by a global gene expression analysis, a transcriptomic analysis, we were able to document it by using all kinds of microbe or parasite or bacteria like Listeria, but we use schistosoma, leishmania, or product from these microbe. And in every case, the IL-2 gene was transcribed, but only on a very, very short window. So between 4 and 8 hours, or even less, there was an induction of transcription, but this was shut down, which is not the case for other cytokines like IL-12. So these tell us that kinetic cytokine production is not something that will occur will be sustained, but can be done on a very short window time frame. And of course, we also showed that this was not only at the transcription level, but was also at the protein level. And in vitro, you could get up to 6,000 picogram per ml, which is more than what a T cell can produce in this autocrine loop. So this time frame is very important because, as I will show you later, it could be that this early IL-2 production might be instrumental in the activation of and the polarization of T cells. But if we look at different cytokines, for instance, IL-6, this has a completely different kinetic, and it will, the transcription will start and go up instead of being shut down like in this case. And this was true for bacteria like Bordetella and most of the microbial world that we have tested. I just want to show you another example of how precise is the sensitivity and the fine-tuning of dendritic cells. We have, for instance, provided to dendritic cells Leishmania mexicana in the form of a mastigote, promastigote, or a mutant, the LPG, which is missing this glycan core. And then we did a transcriptional analysis, and among all the cytokine genes, these are interferon-inducible, but the cytokine genes, the difference between the LPG positive and LPG negative parasites is really into few cytokine, including IL-2, which is completely shut down in transcription when we use the mutant. Again, this shows how recognition is fine-tuned. And when we started to look into sterile signals that, as you know, are also very important danger signals, like, for instance, monosodium urate, uric acid, which is a ligand of the inflammasome, NALP3, we have found that also sterile signals can induce IL-2 production by dendritic cells, and that the synergy with microbial signal, for instance, MDP, which is muramyl dipeptide, and MSU will induce even a higher amount of IL-2. So the amount of IL-2, we believe, matters, and we have found that among the microbial species that we have looked at, the fungi are the one that triggers the highest level of IL-2 expression. And we know that the fungi are a very nice system to study immunity, because depending if they are present as spores or as hyphae, they of course can become. We can undergo aspergillosis, for instance, but only when one of these forms is produced. So if we treat dendritic cells with either hyphae or beta-glucan particles that are particles made only of very pure beta-glucan, and we look into the cytokine genes that are induced, the one that is the most highly induced is IL-2. And this is quite impressive compared to LPS, LPS treatment. And we know that these fungal cell wall components like the beta-glucan, they can be exposed even more when the conidia starts to be a swollen conidia. And we have found that compared to conidia, the swollen conidia and the hyphae will induce very different amount. It seems like there is a kind of IL-2 dose dependent in response to the beta-glucan. And even more interesting, if we were comparing the induction of IL-2 when using either soluble beta-glucan, the PGG, or the particle of beta-glucan, so the same molecular form, but in different physical form. This is a particle and this is soluble. The dendritic cells also discriminate between these two forms, even if the receptor and the molecule is the same. So there is a lot of complexity behind all these that dendritic cells can sense and can discriminate. And we know that the beta-glucan particles triggers SYK and the pathway that I just described. Zimosan does probably similar things. But beta-glucan, a soluble beta-glucan, does not trigger any IL-2 and FET activation and IL-2 production. So all these led us to look into more detail on the amount of IL-2 produced, and we are presently trying to really look into Treg cells, which, as you know, express the CD25 and are very sensitive to very little amount of IL-2. And we know that IL-2 is required for Treg activation, and that in the IL-2 knockout mice, there are no Treg, and there is systemic autoimmunity. So there is a lot that we can learn in the near future, but to go from the bench to the bed, we were very pleased to read this article, which is the first trial published in New England Journal of Medicine last year, the first trial using low doses of IL-2 in GVH host disease. And they have been able to document very nicely in this phase 1/2 trial that indeed the Treg cell response after 8 weeks of daily low dose of IL-2 was really quite impressive. So the CD25 cells, positive cells, were going up, and the disease was doing better. So there is a regulation of probably secretion of IL-2 by dendritic cells, which is tightly organized. And what we now appreciate is that this IL-2 can be uptaken by either the soluble receptors of IL-2 that are produced in large amount, and we find IL-2 receptors in the serum of several diseases, and then we can also see by confocal that this IL-2 can be reuptake also on the IL-2 receptor and can be transpresented to T cells. So probably this is something that has been shown recently by this group, where they have looked at the role of this IL-2 transpresentation in human dendritic cells, and they have shown that this leads to T cell activation through this monoclonal antibody therapy. So we would like to propose a model that we are presently testing. We have generated an IL-2 flox mouse, and we have bred with CD11C Cree, and from these mice we will be able to shut off these early IL-2 produced by dendritic cells and formally prove that indeed in the absence or presence of this early IL-2, maybe the T cell activation might be affected, and in particular Treg cells that are the ones that we are most interested on. And I tried to really present knowledge in the process of being created, as it is stated at the Collège de France, and I would like also to acknowledge the people that did most of this work. Teresa Zelande for with the fungi, and Jan Frick, but also Francesca Granucci at Milano Bicocca. And here you can see that there is two people here, Philippe and myself, and Philippe has been really instrumental to chair SIGN that now has 200 people, and it's a very nice institute, and I wish to thank again Philippe for all his effort in promoting and driving this initiative. Thank you very much.